Welcome back to this next video, and this is the part 2 on the Clostridium perfringens. If I gave you a recap of the previous video, I've told you that C. perfringens, they are one of the most common causes of the food poisoning, and they are also responsible for causing other diseases like the gas gangrene. And the specific name Perfringens, that is derived from the Latin, this per mean through and the fringo mean burst, which actually refers to the disruption of the tissue that occurred during the gas gangrene. The biochemical properties of the Clostridium perfringens were that they were rod shaped, they were gram positive, and they were having a capsule. They were endospore forming bacteria, they were anaerobic, and they were able to tolerate the traces of oxygen by having the ability to produce the superoxide dismutase. They were catalase and oxidase negative, and the C. perfringens, that was non-motile. And the C. perfringens, it actually shows a double zone of the beta hemolysis, in which you actually see the beta hemolysis on the interior, the complete hemolysis, and on the outer side you actually see these partial hemolysis. Then we discussed about the gas gangrene in detail, that this gas gangrene is actually produced by one of the important toxin produced by the C. perfringens, which is known as the α-toxin. This was just an image where the C. perfringens was causing the gas gangrene. And we discussed in detail about the function of the α-toxin. That α-toxin, they have got the phospholipase C and the sphingomyelinase activity. By that they have the ability to hydrolyze the phosphatidylcholine and the sphingomyelin, which are two of the important components of the eukaryotic cell membrane. And by degrading the sphingomyelin, that was actually affecting the transfer of the message from the brain to the other parts of the body. The α-toxin, that was also responsible for hindering the immune response, and they were also responsible for relocation of the platelet fibrinogen receptor from the inside of the platelet to its outer membrane, which was actually a byproduct when you degrade the phosphatidylcholine. One of the byproduct was the diacylglycerol, and this diacylglycerol was actually responsible for this relocation of the receptor, which were making these platelet aggregates. And these platelet aggregates, they were actually responsible for elevated vascular permeability and hence the buildup of the fluid in the tissue, which was referring to the edema. In this particular video, I want to focus on some other virulence factor of the Clostridium perfringens. So one of the other important virulence factor, that is known as the Perfringolysin O, or for short that is known as the PFO. This is Perfringolysin O. Now this Perfringolysin O, or the PFO, that is actually secreted by the bacteria as a water-soluble monomer, and they can only recognize and bind to the cholesterol in the plasma membrane of the host. Now this cholesterol, it is one of the most common component of the animal cells, and this PFO, that actually requires this cholesterol to perform its function. So the first thing this PFO do is that it is going to recognize and attach itself to the cholesterol in the membrane. When it's have attached itself to the cholesterol, what it actually do is that it will be going to make these oligomers, and the PFO, they are going to oligomerize at the host cell exterior, and they actually make a small pore complex. When they make these oligomers, the next thing they do is that they are going to insert themselves into the cell membrane by making a very large pore. Now the formation of this large pore, that actually disrupts the cell membrane, which actually leads to the cell lysis and the cell death. Now this PFO as well as the α-toxin, one of the important things they do is that they actually prevent the white blood cells from arriving at the site of infection. Now if they are actually blocking the arrival of the white blood cells, that means your immune system is no more able to fight this particular infection, which actually causes the cell death and the cell lysis that you see in case of the Perfringolysin toxin. Now the alpha-toxin in the Perfringolysin that also promote the production of the adhesion molecules and platelet activation factor. And this adhesion molecule and this platelet activation factor, what they do is that they are going to clog up the host blood vessels and disallow the passage of the white blood cells to the infection site. Now, as the immune system is not able to fight the bacterium at the site of the infection, this toxin actually aids in the decay of the flesh and tissue. Now, if I summarize the whole story for the PFO, the first thing this toxin do is it is going to bind to the cholesterol inside the membrane. Second, it is going to insert itself into the membrane. Then they are going to oligomerize and make these large pores. And when they make these large pores, they are actually going to disrupt the cells. This is actually a Perfringolysin. As you can see over here, initially they are in the monomeric form, but when they bind to the cholesterol, these yellow dots over here, when they bind over here, they are actually going to make these oligomers, and these oligomers are then going for the disruption of the cell membrane of the host cells. Another important virulence factor of the Clostridium perfringens, that is known as the Clostridium perfringens Enterotoxin. But to understand the function of the Clostridium perfringens Enterotoxin, for short we will call it as the CPE, you need to understand the concept of the tight junctions. Now, when we talk about the epithelial cells of the animals, for example, like the human beings, so these epithelial cells are actually the sheet of the cell that provide the interface between masses of cells and a cavity or space. By that I mean that are actually providing an interface for the lumen. Now, the portion of the cell that is exposed to the lumen is called at its apical surface. As you can see over here, if this is the lumen, so the area of the cell that is exposed to this lumen, they are actually known as the apical surface. Now, the rest of the cells, like starting from here, its sides and the base of the cell, that actually make the basolateral surface. Now, what the tight junctions do is that they are actually going to seal the adjacent cells. If this is one cell, this is another cell, so this tight junction just below the apical surface, they are actually going to seal the adjacent epithelial cell in a narrow band just beneath the apical surface. Now, one of the important protein, they are the component of this tight junction, or which are making these tight junctions, they are known as the claudine proteins. There is a network of the claudine proteins and other proteins. They are actually making these tight junctions between the adjacent epithelial cells. Now, what this CPE is, that the CPE is a polypeptide, that means it is a protein, and the molecular weight of the CPE is 35.5 kilodaltons, and it is one of the major virulence factors of the C. perfringens. Now, what it do is the CPE is actually responsible for the disintegration of the tight junctions between the epithelial cells in the gut. As I've told you, this is the tight junction, so the CPE is actually going to disintegrate these tight junctions. Now, the way the CPE work is that it is going to destroy the cell membrane structure of animals by binding to the claudin family proteins, specifically the claudin 3 and the claudin 4. And when they bind to them, they are actually going to remove this claudin 3 and claudin 4. They are going for the disintegration of the claudin 3 and the claudin 4. And when they are disintegrated, actually the tight junctions between the adjacent epithelial cell, that is gone, which is actually causing the diarrhea in the individuals, as you see in case of the food poisoning. Now, if I give you the whole story, how this CPE is going to cause the cell death or the membrane disintegration, that initially the CPE is going to bind to the claudin proteins. As you can see over here, these are the claudin proteins. These will be the tight junctions between the epithelial cells. So these claudin proteins, the CPE is going to bind to them. When they bind to them, they are actually going to make these small complexes. These small complexes are actually nearly 90 kilodaltons. Now when they make these small complexes, then 6 of these small complexes, they combine with each other, they make these oligomeric structure. And when they make this oligomeric structure, this oligomeric structure is known as the pre-pore, and it is also known as the CH1. If you talk about the molecular weight of this pre-pore, that is actually 450 kilodalton, because you are actually combining 6 of these small complexes with each other. Now what this complex do, when this pre-pore that is formed, this pre-pore is actually going to insert itself into the cell membrane, and when it insert itself into the cell membrane, it is actually causing the influx of the calcium into the cells. Now initially the concentration of the calcium inside the cell, that is low. The concentration of the calcium outside the cell, that is high. That means that when they find pore like this, the calcium is going to move into the cell, so the concentration of the calcium that is going to increase inside the cell. When the calcium enters the cell, what it do is that it is going to activate this particular protein, which is known as the calpain protein, and this calpain protein is going to attach itself to the mitochondria, and when it attaches itself to the mitochondria, this mitochondria is going to release the cytochrome C into the cytoplasm of this epithelial cell. Now when the cytochrome C that is released into the cytoplasm of the cell, that is going to activate the caspases, the special class of the proteins that is responsible for the process of the apoptosis. So when the calcium it moves inside the cell, they are going to activate the calpain, and the calpain is going to release the cytochrome C into the cytoplasm. That release of the cytochrome C is going to cause the activation of the caspase 3, which actually lead to the apoptosis or the cell death. In other cases, how the necrosis that is caused, that is still not known, but some of the authors, they do say that this high concentration of the calcium, when they activate the calpain protein, they can also lead to the necrosis. Whatever the case is, in case of the apoptosis or in the case of the necrosis, there will be the cell death, and when there is cell death, that is going to cause the diarrhea in the individual. So if I summarize the whole story, the CPE is going to bind to the claudin protein, making these small complexes. These small complexes are going to make these oligomers, these pre-pore, which is going to allow the entry of the calcium into the cells, causing the activation of the calpain, which will be responsible for the release of the cytochrome C into the cytoplasm, and hence apoptosis, and hence death of the cells. If you talk about the diagnosis of the Clostridium perfringens, so one of the technique that you can utilize is you can go for the histological specimens. If you talk about the patient of the gas gangrene, in those particular case, what you do is that you are going for the aseptically, you are going to collect the sample aseptically from the infected individual. You then go for the microscopic examination. The spelling is not right. This is not examination. This is examination. Sorry for that. So the in the histological specimen, you are going to collect the specimen aseptically so that it is not contaminated by any other unwanted microorganisms. Then you are going to go for the microscopic examination when you grow them on the culture media under the anaerobic condition. And when you culture them on the media under the aerobic condition, and when there is growth, you can actually go for the microscopic examination and other biochemical tests that I'll tell you in a while. Another sample that you can get is from the wound exudates. One of the important thing to keep in mind about the wound exudate is that you should take the sample from a deeper area where infection seems to be pronounced. So if you are going to take the sample from the upper part, it may be contaminated with other bacteria, or it may not give you the true results. So in case of the wound exudates, you should go for the sample from a deeper area where infection seems to be pronounced. In both of the cases, if you are going to take the histological specimen or the wound exudates after the growth on the culture media, when you go for the microscopic examination, they are actually the gram-positive rods, they are gram-positive bacilli. The other biochemical properties of the Clostridium perfringens is that they are non-motile, they are capsulated, and they are the spore formers. So these are just some of the properties which can help you in the identification of the Clostridium perfringens from the histological or from the wound samples. When you talk about the cultures, the culture media, the important culture media that is utilized for the growth of the Clostridium perfringens, that is known as the Robertson cooked meat medium. And if the sample is positive, and by that I mean if the Clostridium perfringens is there in the sample, and if you grow them on this particular media, there will be blackening of meat which is present in this particular media. And this blackening of it is actually due to the production of the H2S and the ammonia gas by the Clostridium perfringens. If you are going to grow them on the blood agar medium, as I've told you in the previous video, they are going to give you the double zone of the beta hemolysis. Now the Clostridium perfringens, it can cause variety of the diseases. As I've told you, it can cause the food poisoning in the human beings, but it can also cause the gas gangrenes in the human beings. So in case of the gas gangrenes, the symptoms, they are usually the edema, which is actually the buildup of the fluid in the tissue. There will be necrosis of the tissue, which you can call it is the myonecrosis, as I've told you in the last video. The foul smelling discharge is there, the discharge that actually smells like it have got a very foul smell. There can be fever, there can be renal failure, and in severe case it can lead to the death of the individual, or in normal cases, by that I mean in cases of the gas gangrene, there is death of the tissue. When you talk about the food poisoning, which is actually caused by the toxins of the Clostridium perfringens, so diarrhea and abdominal cramps, they are usually there, usually without vomiting or fever, in 7 to 30 hours after the intake of the contaminated food. Now the illness that suddenly begins, and it actually lasts only 1 to 2 days when you talk about the food poisoning, not about the gas gangrene. If you talk about the treatment, if you talk about the treatment of the gas gangrenes, you have to use the antibiotics, and you have to go for surgery to remove all the dead and the infected tissue. Now, if the gas gangrene that is suspected, the treatment must begin immediately, and the first thing you have to do is that you have to go for high doses of the antibiotics, typically the penicillin and the clindamycin, they are given. Secondly, all the dead and the infected tissue that is removed surgically, and in about 1 of 5 people with gas gangrenes in the limb, that actually requires the amputation. But the amputation means the removal of the limb, because all of the limb, if that is dead, that can cause problem in the other body parts. So usually what the doctors do is that they go for the amputation if they are sure that the limb that has been totally affected due to the gas gangrene, and that is no more of use. The third thing you have to do is you have to clean the wounds thoroughly, so that the Clostridium perfringens do not spread to the nearby areas. Excuse me. Another important thing is when you talk about the gas gangrenes, you have to go for the treatment in high pressure oxygen, which is known as the hyperbaric oxygen. Now these high pressure, these hyperbaric oxygen chambers, they are also helpful, but such chambers, they are not always readily available. So what you do in the high pressure oxygen, that you are going to provide the oxygen having a pressure higher than the normal air, and when you provide the high pressure, that actually stop the growth of the Clostridium perfringens, because they are anaerobic, and the oxygen, the high concentration of the oxygen, that is toxic to them. So when you treat the individual with high pressure oxygen in the hyperbaric oxygen chamber, that can actually stop the growth of the Clostridium perfringens. When you talk about the food poisoning caused by the C. perfringens, so there is no specific treatment for C. perfringens infection, because that is caused by the toxin and not by the microorganism itself. Now dehydration can be prevented or treated via oral rehydration. As I've told you, there is death of the epithelial cells, so there is diarrhea, and there is loss of the electrolytes. So the dehydration that can be treated via the oral rehydration. or in severe cases intravenous fluid and electrolyte replacement they should be followed. Now antibiotics are usually not recommended in case of the food poisoning caused by the Perfringens, because it is the toxin that is causing the disease and not the organism itself. So the antibiotics, they are not usually recommended in case of the food poisoning caused by the Clostridium perfringens. Now, one of the interesting thing is that you can actually prevent the cases of the Clostridium perfringens if you follow some of the precautionary measures. So the first thing is, if you want to prevent the infections of the Clostridium perfringens, that you have to cook and keep the food at the correct temperatures. Food like especially the roast of beef, of poultry, that should be cooked to a safe temperature. That means they should be properly cooked, and when they are properly cooked at safe temperature, they should be kept at 160 degrees centigrade or warmer, or 4.4 degrees centigrade or cooler. Because if you are keeping the food warmer than 60 degrees centigrade, or if you are keeping the food cooler than the 4.4 degrees centigrade, both of these temperatures, they usually do not allow the growth of the Clostridium perfringens and hence the production of the toxins. Now these temperatures, they actually prevent the growth of the bacteria that may have survived the cooking. And meat dishes that should be served hot within 2 hours after cooking. And if you do not serve them within 2 hours, so before serving, you should properly reheat them. Now refrigerate the leftovers and reheat them properly, because the leftover food, that should be refrigerated at 4.4 degrees C or cooler as soon as possible, and no more than 2 hours after preparation. Now it is very okay to put the hot food directly into the refrigerator so as to prevent the growth of the Clostridium perfringens and hence the production of the toxins. Now large pots of food, such as the soups or the large cuts of meat, that should be divided into small quantities for refrigeration, so they will cool quickly enough to prevent bacteria from growing. If you are keeping the large soups, like the soup in a very large quantity in very large parts of the food into the fridge, there might be chances for the bacteria to survive. But if you split them into small quantities, that actually they will be cooled very quickly and they are going to prevent the bacteria from growing. Now the leftover should be reheated to at least 74 degrees centigrade before serving. If there is leftover food, you have refrigerated it, so before serving, that should be heated at least to 74 degrees C or higher. Now when in doubt, throw out the food. Now the food, they have dangerous bacteria in them, may not taste, smell or look different. But if you doubt it, any food that has been left out too long, that may be dangerous to eat, even if it's look okay. So when you are in doubt, you should throw that particular food and you should not consume that. So if you like the video, please subscribe to my channel, hit the like button and share it with your friends.